Today's topic is DNA as genetic material. The last class we studied how the DNA act as a genetic material by the experiments of Griffith and Avery. So these two scientists they are going to explain how the genetic material is act as a how the DNA is act as a genetic material, but they are not properly given the evidences. The genetic material it act as a DNA only, so that proper evidences they are not given. So that only the many number of biologists they are disputed into these two early experiments of the Griffith and the Avery. They believed that proteins only hereditary substances. So they believe the proteins only hereditary substances in these the organisms what they are going to uses into the Griffith and the Avery experiment. That means the bacteriophages. So the bacteria how they are going to transmitting by the proteins only. So they are explained into they are believed into the proteins only the hereditary substance. Why they are believes the proteins only the hereditary substance? What is the reason behind this? Why they are strongly believe the proteins only the hereditary substance instead of the DNA? Means in eukaryotic cells we are taking the cellular contents we are taking equal almost all the equal amount of DNA and protein. The protein substances and the DNA is having into the equal contents they are presented here, and this is the one reason. And other reason, the complexity and variability or diversity of protein. So proteins is able to carry out the information which responsible for the hereditary. So because of these properties of the proteins, that is the variability or diversity and the complexity of the protein, these are able to carried out in the different types of the characters from one generation to next. That means they are acting as a hereditary. So these are the previous or the earlier beliefs in case of DNA as a genetic material. They are not believed that DNA as a, but later on in 1952. So in the 1952, Hershey and Chase. So these are the two scientists. They conducted the experiments on bacteria and the bacteriophage. Taking of the bacteriophage and the bacteria, they conducted the experiments. Hershey and the Chase experiment they are conducted, and they are giving the evidences. So evidences are given. The DNA is only acting as a genetic material here. So whether we are taking into the Hershey and the Chase experiments we are taking means Hershey and Chase experiments. So experiment we are taking means Albert Hershey and Martha Chase. So these are the two scientists in 1952. These are the two scientists. So in the 1952, they conducted the one experiments on bacteria, E. coli bacteria and T2 phase here. So T2 phase. So T2 phase virus it is. So phase virus. Phase virus means so the virus which is going to infected or attached to the bacteria. So those viruses are called as a phase virus. So they identified it. So what they are conducted in their experiments means first of all they are identified these T2 phase viruses are infecting into the E. coli bacteria. So T2 phase viruses are infecting into the E. coli bacteria, and after the infection the infected E. coli bacteria is going to lysis and they are going to producing the more the number of progeny of T2 phages here. That means T2 phase viruses are going to producing by the infection of E. coli. So number of phages are going to generating when T2 phase virus is going to infect it into the E. coli cell. So by using these two organisms, so that is the E. coli and the T phase virus, so they are conducted to they want to prove the Hershey and the Chase, they want to prove the DNA as the genetic material. They are taken to these two. So these two organisms, E. coli and the T2 phages, and they conducted the experiment. So that is the Hershey and the Chase. So phase experiment, T2 phase experiment here, or Hershey and the Chase experiment. So what they conducted in this experiment means, first of all, they are taken to the E. coli and T2 phase. So they are taken to these two organisms. So a common properties of the DNA and the proteins, we know that. So proteins are contain sulfur here. So sulfur is presented into the some of the proteins we are taking. DNA contains all the DNA, all the nucleic acids are contains phosphates. So phosphate molecules are presented into the nucleic acid. Like that, the DNA also a nucleic acid contains into the phosphate. So that only they are taking the isotopes of the sulfur and the isotopes of the phosphate. The sulfur isotopes S35 they are taken, and the phosphate isotope P32 they are taken. So these two isotopes are they are taken to conduct the experiments on the E. coli and the T2 phase viruses we are taking. So how they are going to conduct it into the experiment means, so initially they are taken the radio labeled, so these bacteria and the phages they are taken here. First they are radio labeled into the bacteria phase. E. coli bacteria they are taken without any labeling phase. First they are taken to the T2 phase. So T2 phase virus, which are radio labeled into the two different ways here. So what are those two different ways? One, T2 phase virus is radio labeled, it is radio labeled in their head, or their protein coat is going to radio labeled by the S35. So it is going to radio label. The inner genetic material is as usual here. So it is into the red color. They are radio labeled. So with the isotopes, S35 isotopes, their protein code. So this is the one type of the T2 phase viruses here. Another T2 phase viruses, they are taken. So for our differentiation, we are going to the bacteria, so we are going to differentiate the bacteriophage virus. So this is another phase virus, it is having the radio labeled nucleic acid. So this is into the radio labeled nucleic acid. So wherever here the thicker lines are representing into the, this is into the radio label. Here the thicker lines are going to representing the nucleic acid is a radio label. Here the nucleic acid is a normal here. So like this nucleic acid is P32. It is radio labeled by the P32. It is having in the red color isotopes. The red color DNA is presented into the P32. Radio label is present. So when these two T2 phase viruses and along with these two radio labeled and the non-radio labeled E. coli cells, radio labeled bacteriophages and the non-radio labeled E. coli cells, all those are going to mix. So when they are going to mixing into the all these radio labeled and the non-radio labeled E. coli and the phase viruses, or it is happened means the phase viruses are going to attack on the bacterial cell. So this is into the bacterial cell, E. coli bacteria. It is the E. coli bacteria. When these phase viruses are attacks on bacterial cell, this is also attacks on the bacterial cell. So it is attacks on the bacterial cell. Immediately the bacteriophage is introducing some materials into the E. coli cell or bacteria. So to track into the separately, either the protein is going to introducing into the bacterial cell or either the DNA is going to introducing into the bacterial cell, to tracking into the separately protein and the DNA or the nucleic acid of the phase viruses. So they are conducted into the radio labeled isotopes are taken. After the infection of the bacteriophage with the E. coli cell, they conducted a gentle agitation, that is a centrifugation. They are conducted a gentle centrifugation. They are conducted and they are separated into these attached bacteriophages from the bacteria. Later they are identified that the infected bacterial cell is received only the radio labeled DNA only. So it is received into the infected cells are they are received. They are identified into the infected cells or the cells of the E. coli bacteria. So they are identified only the radio labeled P32. So in any case we are taking here P32 radio labeled molecules only they are enters into the bacterial cells. They are not identified into the S35 so cells into the inside the bacterial cells here. So by this differentiation they concluded that from bacteriophage to the bacteria to producing into their next generation, only the DNA molecules are going to transmitting, not into the protein or the protein molecules. So based on this experiment, so they concluded that DNA is acting as a genetic material here. So this is a supporting experiment towards into the DNA acting as a genetic material. For the experiment, it gives the supports to the experiments of the Griffith and the other transformation experiments we are taking. So like this, they are concluded that DNA as a genetic material. Having identified into the nucleic acids as DNA and RNA. That means after the identification DNA and RNA as a nucleic acid or genetic material, they are started the studies on the structure of these nucleic acids, that means the DNA and the RNA here. So whether we are taking in organism nucleic acid, DNA, either the DNA and RNA, how they are going to arrange means they are arranged as a polymer structure here, polymer chain structure. So it forming into the polymer chain structure. That means DNA and RNA, they are going to forming number of units are linked and forming into a polymer complex chain structures are formed. So what are these unit linked which are linked into this polymer chain structure either into the DNA and the RNA means generally they are nothing but as a nucleotides here. Nucleotides. So nucleotides are presented, a number of nucleotides together. So they are interlinking and forming into a polymerized chain structure is going to forming. So that structure, that condensed polymerized chain structure is called as a nucleic acid. Either it may be the DNA or it may be the RNA. So both are forming the polymerized chain structures. Whether you are taking into the components of nucleotide structure you are taking. And what are the components of these nucleotides? That means sugar, nitrogen base, and the phosphate. So these are the components of nucleotides. Sugar, nitrogen base, and the phosphate ions. So one by one you can be study. First of all, sugars. Sugar. So the sugar molecules. One sugar molecule is presented. This is pentose sugar. Pentose sugar molecule. So pentose sugar molecule. Here the specification is there between into the DNA and the RNA. The sugar molecule, the structure of the sugar molecule. Whether you are taking the DNA is forming deoxyribose sugar. Deoxyribose sugar, and RNA is forming so ribose sugar molecule. Actually this ribose sugar is a pentose sugar, pentamer here, that is a 5 carbon. So 5 carbon ring structure is forming. So whether you are taking into the deoxyribose sugar, their structure. So this is into the deoxyribose sugar structure. So this is the deoxyribose sugar structure here. So this is the deoxyribose sugar structure. This is the deoxyribose sugar. Whether you are taking the ribose sugar structure, it is also pentose sugar only. Only the differentiation is one oxygen molecule. So you are taking HOH. So here only the differentiation is the second portion of the ribose sugar. Here the deoxyribose, DNA ribose sugar we are taking, oxygen is absent in the second portion. In the RNA, the second portion oxygen is present. This is only the difference between the sugar the pentose sugar molecule between the DNA and the RNA. So this is the one component in the nucleotide we are taking. And the second component we are taking is nitrogen base and the phosphate ions. So nitrogen bases we are taking. Nitrogen base. So nitrogen bases we are taking. There are the two forms of the nitrogen bases are forming either in the DNA or RNA. Totally each DNA contains 4 types of the nitrogen bases and RNA contains the 4 types of the nitrogen bases. They are adenine, guanine, cytosine and thymine. So these are the 4 nitrogen bases in case of DNA, and in case of the RNA, adenine, guanine, cytosine and the thymine, and the uracil. So this is the basic difference between the nitrogen bases of DNA and the RNA. The thymine is unique for DNA. The uracil is unique for RNA we are taking. Here in these 4 nitrogen bases, cytosine and the thymine, adenine and the guanine. Adenine and the guanine is forming by the 2 ring structures. 2 ring structures. They are going to forming 2 ring structures. So this is the 2 ring structure is presented. 2 ring structure is forming here. 2 ring structure is forming. That is purine ring. This is called as a 2 ring structure. 2 carbon ring structures are forming in the adenine and the guanine. So these 2 rings are called as a purine rings. So these are called as a purines. So these 2 nucleotides are called as a purines here. And then we are taking the cytosine and the thymine, or the cytosine or the uracil is forming a single ring structure is going to forming. That is a 6-membered ring structure is going to forming. NH2 NH. So this is into the pyrimidine ring structure. So purine rings and the pyrimidine rings, like this purine and the pyrimidine. So these 2 rings are going to the basic structures of the differentiation between the adenine and the cytosine, guanine and the thymine we are taking like this. So these 2 are forming these 2 rings, and these 2 are forming so these are called as a purines and these are called as a pyrimidines here. Purines and the pyrimidines in the DNA, like that as a purines and the pyrimidines in the RNA here. So like these 2 purines, 2 pyrimidines, nitrogen bases are going to forming here. And these nitrogen bases, so basicity, that is nitrogen bases are going to forming. So basic nature of this nucleotide sequence here. Later we are taking phosphate here. Later we are taking phosphate. So phosphate is derivative of phosphoric acid. Phosphoric acid, H3PO4. So the derivative of the phosphoric acid. Phosphate group is nothing but as a derivative. Phosphate group ion. So the phosphate functional group is the derivative of phosphoric acid. From this phosphoric acid, phosphate functional group is going to forming, and it contains 4 oxygens here. So it is attaches with the 4 oxygens. Actually it contains the 3 oxygens, in that the 2 is the stable and 1 is into the negatively charged here. So later the phosphate ion going to forming a complex. The polymerization is occurs in case of nucleotides with the help of phosphate molecule only. So this phosphate ion is going to releases the H plus ions. H plus ion. So because of the releasing of the H plus ion, the nucleotides are getting the acidic nature. So that only we are giving the nucleic acid here. Nucleic acids. While containing into the nitrogen bases also, the overall charges or the overall pHs of the total nucleotides we are taking means acidic pHs they are going to form. Acidic conditions are, so these are called as a nucleic acid. The polarity is also going to, so the polarity, the pHs, not polarity, the pHs are going to determined by the, so nucleic acids with the help of, so phosphate ions only. Phosphates are having the one negative charge and the two stable oxygen groups. With the phosphodiester bonds, it is going to forming into the structure, nucleotide structure we are taking. So whether the nucleotide structure we are taking, here already we discussed the ribose sugar, either in the case of the DNA or either in the case of the RNA, the ribose sugar is going to attaches, all these molecules are going to attaches into the ribose sugar and forming the polypeptidal chain structure here. So how the polypeptidal chain structure and the what is the arrangement of the, so these components of the nucleotides one by one we are going to discuss. So the structure, how these sugar molecules, nitrogen bases and the phosphate ions, how they are going to combining and forming the polypeptidal that is a polymer chain of, so nucleotides means, so initially we are taking, so this is the sugar molecule. The sugar molecule is having 1, 2, 3 and the 4, 5 carbons are presented. At the third carbon OH group, so already we discussed here one OH group is there. That OH group is reacted with the phosphate ion. The phosphate ion already three stable oxygen, three oxygen molecules are present in that. The two is the stable, one is going to acting into the this OH and releasing into the oxygen. Oxygen is going to liberate. H plus ions are going to live. So that only it is formed the acidity. Acidic condition is forming in the genetic material. So like this, the third portion of the OH is reacted the phosphate and forming the phosphodiester. This is this side the one oxygen, this side the another oxygen. The phosphate is going to form the phosphate diester bonds are forming with the ribose sugars in the both the sides. And later the sugar molecule is attaches the another side, the first carbon of the sugar molecule is attaches with their nitrogen bases here. Nitrogen base structures already we know that purines and the pyrimidines. Adenine and the guanine is act as a purine. These are forming by the two ring structure. For example, this is the adenine here. The adenine purine structure, purine ring is presented. It is going to attaches with the thymine. It is going to attaches with the thymine. So the adenine is always combines with the thymine. Guanine is always combines with the cytosine only. Why? And how it is going to combines with the adenine is always combines with the thymine, the guanine is always combines with the cytosine. Means adenine with the help of two hydrogen bonds. These are the two hydrogen bonds. So between into the adenine and the thymine. So there is forming of the another hydrogen bond, it is not possible here. So based on their structural configuration, these are able to producing the two hydrogen bonds between the adenine and the thymine. So they are forming into the complementary. And the guanine, the cytosine is able to produce the three hydrogen bonds because of their structure only. This is also here the reason is why these are forming the two, why these are forming the three. It's based on their structure only. These are forming into the three hydrogen bonds between the guanine and the cytosine here. So like this, adenine is always binds with the thymine, guanine is always binds with the cytosine here. So like the ribose sugars are attaches with the nitrogen base. One side it is attaches the phosphate ion and the first carbon place it is attaches with the So nitrogen base, and the fifth carbon, so this is entirely into the one nucleotide. So nitrogen base, sugar, and the one phosphate ion is comes under the one nucleotide. So this nucleotide is going to combines with the another nucleotide. It is going to attaches with the another nucleotide. There 3 prime end here. So 3 prime and the 5 prime means here the fifth carbon of sugar molecule is going to attaches. So already the fifth carbon of the sugar molecule is stable, and the OH group, whenever the number of polymers are going to adding means where they are going to adding means the third 3 OH group, the third place of the OH group of the sugar only they are going to attach is the another nucleotides. Continuous number of nucleotides are going to adding into the third position of the sugar molecule. So this is called as a 3 prime and this is called as a 5. Based on the position of the carbons of the sugar, we are giving into the name of the 5 prime and the 3 prime. Always the 3 prime directions only number of nucleotides are going to adding here. So like this, one side it is forming into the one strand, DNA strand is going to forming, and the other side it is also forming into the another strand of the DNA. So like this we are taking. So here again it is going to forming another strand of the DNA. But these two strands are not parallel. These are the opposite strands here. Opposite strand means one strand 5 prime to 3 prime. It is presented the 5 prime to 3 prime. Another strand is presented the 3 prime to 5 prime here. So like this, the strands are oppositely they are arranged into the DNA structure here. So this is the arrangement of the components of the nucleotides here. And how these are forming into the chemical natures means here the phosphate ion is continuously releasing into the H plus ions. Whenever the new nucleotide is going to add it in the phosphate group, phosphate functional group, so each time it releases into the one H plus ion and forming the OH minus here on their structure. So like this, number of H plus ions are going to releases into the addition of the number of nucleotides. Total overall the pH values of the nucleotides are becomes into the acidic, and the overall charges into the DNA molecule is a negatively charged is formed. The polarity we are taking towards into the, so fifth carbon we are taking 5 prime is presented towards into the third carbon, it is a 3 prime and it is forming here. So without knowing these two, 5 prime which is the 5 prime and which is a 3 prime, and how the nucleotides where they are going to adding into the new nucleotides where they are going to adding, without understand this one, the further process of the molecular genetics we are not going to able to understand here. So this one is the arrangement. Wilkins and the Franklins. These two scientists are conducting the X-ray diffraction analysis here. X-ray diffractions. So the Wilkin and the Franklin X-ray diffraction analysis, using of the X-ray diffraction analysis of the Wilkins and the Franklin. Later on, Watson and Crick. So these are the two scientists. They are explained the structure of the DNA. Watson-Crick model. They are going to explain how the DNA strands are complementarily. They are going to arrange. Here, the arrangement of the two strands of the DNA always they are following into the one specific group. That is explained by the Chargaffs here. So it is called as a Chargaff rule. Chargaff rule. So what is a Chargaff rule means? Here in DNA or RNA, whatever it may be, DNA we are taking, generally DNA, number of adenines always equal to the thymines and the number of guanines always equal to the cytosine. So that means overall in DNA structure we are taking purines equal to pyrimidines here. So purines are equal to pyramids. Why? Adenine and the guanine is a purine, thymine and the cytosine is a pyrimidine we are taking. That means number of, how many number of adenines are there, that many number of thymines are compulsory they are presented. How many number of guanines are there, that many number of cytosines are presented. So that means always the purines are always equal to number of pyrimidines in a DNA material. So this is called a Chargaff rule. So by the following of the Chargaff rule, the DNA molecules they are forming in the nucleotides. The nucleotides are going to forming in the nucleus of the eukaryotes or nucleons of the prokaryotic structure cell. Only the difference between the RNA and the DNA, here instead of thymine, uracil is presented here. So uracil is presented instead of the thymine, uracil is presented. So the uracil is nothing but as here uracil is means the difference between the molecular structure of the thymine is going to changes by losing of this methane ion. It is going to forming into the uracil structure. So this is only the difference between the RNA and the DNA structure. So this is the structural description and how the DNA is going to act as a genetic material.